Dear participants, good evening all. It's my pleasure to welcome you all to this 15 days internship and certificate course on artificial intelligence and machine learning. So today is going to be our first session. I am Praveen Kumar. I am the managing director of Institute of Innovations. We are located in Tiruvannamalai, and I would like to give some brief introduction about this program and our resource person, Dr. Praveen Kumar. So I myself also Praveen Kumar, and our resource person for this course is also Dr. Praveen Kumar. So before going into the session, I would like to give a brief intro about Dr. Praveen. Dr. Praveen is a biotechnologist from Sri Venkateswara College of Engineering from Chennai. He is a professor. He is a PDRF in IIT Madras. He had completed his bachelor's in biotechnology in the year 2000 and post graduation in the year 2006. He got a doctoral degree from Anna University Chennai in 2020. He is now working as professor in biotechnology from in Sri Venkateswara College of Engineering Chennai. Earlier, he had worked in many institutions, and he is a well-known person to our institute, that is the Institute of Innovations. He has handled somewhat more than 5 to 6 courses from our institution, which we conducted online from the year 2021. So I'm sure that he will be giving you more insight about in this subject area. Welcome you, sir. Welcome, Professor.
Thank you, sir, for the brief introduction. Good evening to you and to all the participants for the first session. Thank you.
Yes, Professor. I'll take another 2 minutes, couple of minutes, Professor.
Yes, Professor.
So dear participants, I would like to give you some 2 to 3 main instructions regarding this course, regarding this Google Meet and other things in this about this course. So actually, this course is for 15 days. We are starting today, and the last day is on 30th of May. So on the daily, we are having the course at 7 to 8 p.m. And I request all the participants to join well in time, that is before 7 p.m. You can join the session from 6:55 p.m. and it will be easy for us to follow all of you in the session. So the course will be ending on 30th. That is on 29th of May, we are having the last lecture session. And on 30th, we are having a small assessment. Regarding this assessment, many of you asked the queries in WhatsApp that there is no passing minimum in this assessment. That is, I mean that there is no pass or fail in this assessment. The only condition is all the participants should attend the assessment to get the certificate. That is our only condition. So without attending the assessment, we could not able to give the certificate. And I would like to give another important instruction also. We have sent the Google Calendar invitation to all the participants' email ID. That is your registered email ID. So I am sharing the model of the Google Calendar notification so that it will be easy for you. I think my screen is visible to you, right? Yeah, it's visible. So I had sent the Google Calendar notification to all the registered participants, and you might be received email like this from our institute official email ID, praveen@innovationinstitute.co.in. If you didn't receive any email like this, you just search your email box. I am putting this email ID in the chat box also here. Just a minute. So we had sent the Google Calendar notification from this email ID, that is praveen@innovationinstitute.co.in. So you might have received a email like this. So in this email, I request all the participants to select yes. Most of the participants has accepted the invitation. If you accept this invitation, if you select the yes, then you can able to join the Google Meet directly when you are clicking the link. I think I am clear. I'll send this message once again to your WhatsApp also later tonight. So the thing is, we had sent a Google Calendar notification. You might have received like a mail like this to your registered email ID. You just select this yes in that email, so that if you select the yes, you can directly join to the Google Meet directly. Or otherwise, you have to give the request daily, and we have to accept your request, so that the joining process will take somewhat some time to join. So if you are not noticing your request, we could not able to accept. So I request all the participants to go to your email and select this yes. And another important thing is, we participants are asking about this WhatsApp group. We are not we not used to create WhatsApp groups regularly, but we will send the WhatsApp broadcast message. So what WhatsApp broadcast message will be received only to the participants who are saved our institute number in your mobile. So I request all of you to save our institute mobile number in your device. So I am putting my institute mobile number here. That is 989488773165. Just save this number in your device, so that you will receive the broadcast messages. So another important thing the participants are asking to me is about the video recordings. We will send the daily video recording on the night of exam. For example, today's video will be sent to you by after 10:30 p.m. and the feedback for today's session also will be sent with the video link. So all the participants are requested to send the feedback. We will notice if there are any critical things in the feedback, we will surely give you some solution for that. So this is what from my side right now. So if you have any queries about this joining the course and other Google meetings, and another important thing is we will be recording all the sessions. So sometimes what happens means, if you switch on the recording, some participant might be automatically removed from the meeting. That is because of the older version of the Google Meet software. So if you are automatically removed from the meeting, don't panic. You just uninstall your older version of Google Meet software and install the new version, so that you will be seamlessly join the session. So if anything happens, don't panic, and please be free to send us WhatsApp messages. Most of the time we could not able to speak over phone, so send the WhatsApp messages. We'll give solution to you. So I think I am clear for now. If you have any queries, you can send the message. I will rectify that after the session. Thank you. Thank you, Professor. You can take over the session, Professor.
Thank you very much, Professor, for providing me one more chance to take on this same course. So I just share my screen, and I think the session is already started recording.
Yes, sir.
Yeah, is it visible, Professor?
It's loading. Yes, Professor. It's visible. Yeah.
Yeah, so thank you very much for a brief introduction. And so today I'm going to take on the certificate course. So this is for the second time I'm taking this course, that is on online internship and certificate course on artificial intelligence and machine learning approaches of drug and vaccine design in cancer. Okay, so actually we have splitted this article. I think now presently I've shared the Word document, isn't it? Is it visible to you? Because I just moved on to the Word by stopping sharing the PPT. Any participant can interact. The participant can interact by unmute. Even
It is visible.
it's visible, Professor. Okay, good. Thank you. And participants, my suggestion is like whatever our chairman of this Institute Innovation of innovations has mentioned that try to include yourself, okay, as a participant by clicking yes in the mail received. Okay, so if and if you click that, then also you can better put this kind of a URL in a private WhatsApp whenever you want to. Even you can log in. Okay, so or you can use the same kind of invitation already provided by him, so that you can join without anticipating me to provide accept during the presentation. Okay, so during the presentation, I will be taking you to the PowerPoint and immediately I take you to the software and illustration. So almost 40% of hands-on experience also can observe, okay, during this kind of a presentation. So there should not be any disturbance, okay. If there is any disturbance, obviously it affects everyone. And I believe for the last time, I think we had only just 248 participants. Still, it's a voluminous. Many participants joined in the same certificate course, and so I just suggested the chairman to just include the same, the director, Dr. Praveen Ramanujan sir, to include the same title. And I think this time we got nearly 300 participants, around 300. Isn't it, Professor? Are you there? I think yesterday I came to know that we had
Professor,
more than.
sorry, I could not able to get you.
How many participants are registered on present?
274, Professor.
Okay, okay. So this is the final updation. Okay, okay. Thank you.
Yeah,
Because
yeah.
even in Tanvas, I came to know that some few participants want to join. I think may not today, tomorrow they'll be joining. Okay.
Okay.
Thank you, Professor. So this is the certificate course, online internship and certificate course on AI and machine learning approaches of drug and vaccine design in cancer. But I'm not going to take much on artificial intelligence and machine learning because I found the participants are from diversified fields. Okay, so many participants before enrolling on participation, they came to know that the BSc, MSc participants, and also from the BTech, MTech background, and even a few MBBS background students, and even I came to know that even from Thailand, Indonesia, and from South Korea, so many international students also asked me what kind of how to join and how to enroll all this. So I think everyone are in the diversified field from the bachelor's degree to the PhD and even postdoctoral fellow participants also enrolled, I believe, because that's what commonly observed in all the certificate courses. So we have designed a course in such a way that to just start from the fundamentals. Okay, so just taking first two sessions on target discovery, basically on biological databases, target identification, modeling, and validation. That is about the protein targets. And the next for 5 days is about the drug design concepts. That is about the methods, drug databases, and the properties, physicochemical properties of the drugs. Okay, which includes even drug likeness and ADMET properties. And the fifth session, okay, that is the third session of drug design is on molecular docking. So I'll be taking you to teach on Autodoc. Okay, so I'll be providing a hands-on experience on Autodoc, how to install and how to work on Autodoc tools. And then we go for the sixth session on analysis of the molecular docking from the Autodoc. So I'll be illustrating using the Discovery Studio visualizer, and also going to take you visual screening method that is through Autodoc PYRX. And then on seventh day, that is the final day of drug design, I'll be taking you on energy minimization protocols and methods, and we also discuss about the MD simulation, especially through GROMACS. And the eighth and ninth sessions are about the systems biology approaches. That is basically I take you on cancer biology, genomics, and epigenetics. And also we go across some kind of a databases, how to use the data. And how about the network pharmacology and pathway analysis? So this I take you to the network databases and illustrate you through Cytoscape 2. And then for the vaccine design, we take the basic, I'm going to take you the basic concepts on reverse vaccinology and various immunological databases like IEDB. And on 11th and 12th day, I'll be taking you on the T cell and B cell epitope vaccine design. Okay, so both theoretical as well as the hands-on experience. And the 13th and 14th day sessions are about the machine learning techniques and applications of AI. So how to develop these tools. Actually, you're going to use all the tools developed from the AI and machine learning, of course. But how to develop such a kind of a tools? Okay, so some kind of an introduction, how the applications are pertain to the precision medicine design. Okay, so how we can take up to that. So all these things we'll be discussing on the last two session, and of course the final session, the 15th day, is on to assess you. But this is not for grading you. This is just for having a recap. Okay, recapitulate what exactly we discussed on the last 14 sessions. So obviously, you're fortunate to receive the recorded videos, even if you miss one or two videos just because of unavoidable circumstances, so you still can able to manage by watching these videos. Okay, so I think I can start my first session on chapter one on biological databases. Myself, I just a professor, Department of Biotechnology from Sri Venkateswara College of Engineering, Professor Tara Research Fellow from IIT Madras, and of course, I'm a PhD supervisor and UC 2.0 winner for the last year. So these are the certificate courses taken for the Institute of Innovation Tiruvannamalai, which is approved by MSME, of course, a part of AACT. Totally I've taken almost 8 programs, 8 certificate courses, okay, including the large and small certificate courses, and this is the 9th course, okay. So I just collaborated and taking these courses successfully from him, and even the participants numbers are increased drastically. Initially, we got nearly 70 to 100, but each and every certificate course, minimum 30 to 50 participants are increased rapidly. Now we can come across the a kind of feedback from the participants. Many participants have provided that feedback in their own LinkedIn profile. One such thing I got from a bioinformatician. Okay, so she has mentioned that it's definitely useful for her in for doing the PhD research. Not only this, even myself, when I gone to the Welltech University, Chennai, an African student who enrolled PhD in the Welltech University was mentioning that whatever he has done, the 80% of the PhD research is pertaining to this 14 days program. Okay, so what I mean is this is not just a introductory course for that. Okay, even you can take this course. Okay, even you can take this course. Okay, up to uh, the research. Okay, so up to doing the research, that like completing the PhD or even the PhD. Now, highlights of the today's presentation. I'll be taking you on the oncology databases, and also I'll be taking you on the sim biology tool from the MATLAB. So, oncology databases. What are the different oncology databases we come across? I think many of them are aware of the protein sequence databases, nucleotide sequence databases. Okay, like if you go to the protein sequence databases, I think I can now take you to the website and I can illustrate you. So this kind of thing you can avoid participants. Like whenever if I want to start continuous, because the 1 hour is not sufficient, it seems. Okay, even myself I can't able to even go beyond. So try to accept the invitation. Okay, sent by the director, so that it will be easy for us. So if you go to the overall biological databases, defaultly anyone know that we go for, if you want to download any sequence databases like protein or nucleotide, we go for NCBI, isn't it? What is NCBI? National Center for Biotechnology Information. And the URL for this is ncba.nlm.nih.gov. Okay, National Labor on Medicine, National Institute of Health, and GOV, we refer to the US government. So if you see this kind of a database, okay, so this is a web resource where you can find many databases, especially the bioinformaticians are well aware that we generally go for downloading the protein sequences, okay, through this protein database. Whatever the protein database I've selected, I think even I can interact with you so that we can make the sessions interesting. Can someone tell me what is the database of NCBI protein? We have a name for NCBI protein, isn't it? So what is the name of NCBI protein? And whatever, so this is the first question. If you know, you can type your answer there. No, it's not a, it's a sequence database, isn't it? So this is not a correct answer. not PDB. Very good. The correct answer is GenPep. Okay. So if I select protein and if I type, if I want to type any protein, can I type TP53? Okay. So I'm just checking the relevant kind of a protein. So we got it as a tumor protein. Okay, if I want to take a Homo sapiens, this is an advanced filter. We can selectively select only the Homo sapiens alone. Nearly 3067 proteins are available for human. Okay, so mostly we are observing the partial protein sequences. Okay, so we got a very small size. So we can go to the next link so that whether can check, we can get a full length protein sequence of TP53. So till now we are getting only the partial sequences. If you want to take a full sequence, you can check a sequence in such a way that you won't get a partial thing. Okay. So default, I can go to the previous one. Any doubts you have? Now itself, I'm getting some kind of messages. So I can select a kind of for downloading the protein sequences. I can download like this. Okay. Now I can take you to the next, the nucleotide. Okay. So whatever shares the madam has mentioned, that is the GenPep for downloading a protein sequence. First, I can open a FASTA format of a protein. Then I can show you a nucleotide. So now I just opening even a partial protein of TP53 of human. Okay. So this is how a FASTA format looks like. Okay, so you'll be having a GenBank or GenPep taxation number, and then this is the locus name, that is the protein name, and the another locus name is the species name. And the second line, you have only the sequence, amino acid sequence of a protein. So the first line is generally the identifiers, database identifiers, and the second line is just the amino acid sequence for a protein. If that is so, how about for a nucleotide? Even if you want to take a nucleotide, fetch a nucleotide for the same kind of a protein, okay, simply you type the same keyword. Okay, if you know the these kind of an accession number, that is AXC or something like that, is for protein, isn't it? If you know for the nucleotide, if you type the accession number, so this is the accession number for the nucleotide. So the size, what they mentioned, is 123 base pairs linear DNA for the exon 5 and partial series. You can also check for entire nucleotide sequence and you can download the FASTA format. Now, presently, if I want to download the take the FASTA format, I'm simply clicking the sequence. Okay, so now you'll be getting the sequence like this. So this is how you can able to take the sequence. And if you want to save this kind of a sequences, I better suggest you to just copy it and use a kind of a notepad. Okay, don't use any other kind of editor like WordPad or the Microsoft Word, because it confuses the format. So it's better to save these kind of documents in 1 minute. Yeah, so I got a question actually, how to understand that which one is a complete sequence. So if you want to know which is a complete sequence, okay, so you should get like you sh whatever input you're going to get for a protein or a nucleotide, that should be full. Okay, that should not be any something like a partial or exon. So you can find partial in protein, and for the nucleotides, you may get these kind of an exons or partial CDS. So it should not be like this. Okay, the hyperlink, whatever you're going to click, okay, that should be full. Okay, then only you can able to. So until you get this kind of a full sequence, so how many sequences you have? Totally 4,224. You move page by page. Okay, you move page by page, and you try to find the full sequence. Okay, so this is how we can able to fetch the sequence, the entire protein or nucleotide sequence. If you see this partial nucleotide sequence, like however you found in GenPep, so this is the GenBank. I think one participant has unmuted. So if I ask you to answer, you can unmute and answer, or else Press kindly mute. Yeah, thank you. Okay, so just opening this kind of nucleotide sequence in FASTA format. So initially, you'll be getting this greater sign. Actually, this is a kind of a pipeline. Okay, according to the Unix command, and this is actually an accession number for the TP53 Homo sapiens, and of course, this is a locus name denoting the species name, and of course, this is a second locus name denoting the name of the gene, and the remaining is about the details of the gene, whether it's a full gene or is it a partial. Okay, so all this information are denoted in the FASTA format. Okay, so this is the general database we choose. Okay, why we choose this kind of database that is why we choose this kind of nucleotide or protein because we have large numbers, okay, large voluminous statements present in this. And of course, this is the National Library of Medicine maintained from the US government. Okay, so this is the largest database, publicly domain databases, not only for the protein and nucleotide sequences, but we also have data, okay, related to clinical variants, like whatever the doctor has taken data the clinicians have taken from the patients. Okay, all these data are deposited in ClinVar, and the sample related kind of an information, biosamples information are found here by projects, by collections, genome, the entire genome that is. But we won't get multiple genomes, but for a species, you get only one genome deposited in this NCVA. Okay, and related to microarray microarrays data, so you go get in the form of gene expression, isn't it? So this is actual gene expression omnibus. So if you want to get data in the form of a data sets for a cluster of genes, okay, can able to download from here, okay, similar genes. And if you want to download the just the profiles of a gene, you can download from here. And later to an important database is the PubMed. If you want to download any biological literatures, okay, biotechnology literatures, we go for PubMed. And there is also one more literature called Medline. Okay, so mentioned. So actually, these are some of the publications related literatures are available there in these databases. Okay, if you want to download the 3D structure of a compound, 2D structure of a compound, better use this database called PubChem compound. And for single nucleotide polymorphism, okay, so related to that kind of DNA fragments, you can better take it from the SNP and related to taxonomy. And if you want to download even the 3D structure of proteins, okay, so you can able to download from this. Okay, so anyone know about the name of the 3D structure protein database available in NCBA? This is the second question. So for the sequence, the madam has mentioned it is a chain pep. And for nucleotide, we call that as a GenBank, the name of the database. And how about for the structure database present in NCB? Can someone tell me the name of structure database? No. Yes, someone answered right. MMDB. Yes, again, the madam has answered right. MMDB is the correct answer. Okay, molecular modeling database. So this is the database. Okay, if you simply select the kind of a structure, and if you type a kind of a protein, okay, protein keyword, you can type any protein, whatever you want. If you know about the insulin, okay, that's the smallest protein, isn't it? So if you want to download the protein, you can type that insulin, or you can type whatever protein you know. Okay, when I type that kind of protein, I got this kind of a 3D structure. So if you want to view this 3D structure in this kind of ICN 3D viewer, you can click here. Or if you want to download the structure, even that is possible with you by clicking here. Okay, so now I just taking you to the URL. Okay, I'm taking you to the P53 protein. Okay, so this is something about the 3D structure of the P53. Okay, so you can find more annotation of the 3D structure data is provided here. This is actually DNA binding protein. Okay, so how about the DNA, and what is the size, and how about the protein, and what is the size, and how about the motifs present in the protein? Okay, so every information, the annotation, you can able to find from here. And if you want to view it in 3D view, you can click here full feature 3D viewer. It takes you to the new window, and it explores you to check even the XYZ coordinates or wherever you're going to point. Not only that, even it can able to color according to however you want to make it out. Okay, like if you want to color based on the secondary or based on the hydrophobicity or any other physical chemical parameters, we can able to interact well with this kind of a tool. Okay, so we also have Java applet, some tools even for visualizing this in few more online tools. Okay, now let me take you to the next. So I think now you uh, yeah, so it can accommodate your doubts. Okay, so I plan to take nearly 40 minutes theory and last 20 minutes practical. So I'm not going to take the entire 20. I take 15 to 20 minutes. So last 5 minutes I can keep you in the form of a question round. You can ask me whatever doubts you have. Okay, we sharp startly at 7 o'clock and complete sharply at 8 o'clock. So I ensure that I try to reduce the theory content as much as possible and try to uh, boost more with hands-on experience on my tools. Okay, let me take you to the tool once again. So this is the database, and it directs you to the kind of a tool. Okay, so that is from the MMTV database. I've taken to this kind of a 3D visualizer tool, and I'm just showing this kind of a routine with the kind of a detailed annotations. Okay, even if you want to find more annotated information with respect to coloring, okay, even you can select it and you can check according. Okay, if you want to download, okay, if you want to search similar structure, everything is available here. The more options are available. So even I suggest you whatever I'm going to teach you, I can able to teach you whatever I teach you. You have to practice it on the same day. Okay, that is after 8 o'clock. You can go and practice. Then only you can have an idea. Because just for observation itself, you take much time. That's because you have to understand, isn't it? So I take you leisurely each and every tracking of data in the database. Okay, now let me complete about this kind of an NCBA. So I have taken you to this National Center for Biotechnology Information. Initially, I showed you how to visualize the nucleotide sequence or a protein sequence from the database, and now I also taken you to how to visualize even the 3D structure of a protein from the NCBA, that is through molecular modeling database. Now I take you to the another database called PubChem compound. Okay, if you want to download any 3D structure of a drug or any plant compounds, okay, so this on molecules, okay, the natural molecules or the synthetic molecules are available in this kind of a database. Plenty of molecules are there. Even it's not a must to type just the compound name. Even typing a disease name, like for example, SARS-CoV-2. Okay, there are we have data sets available here. So if you go to the data set, obviously take you to the list of drugs or compounds used for treating that SARS-CoV. Okay, so there are so many key fields, okay, keywords to fetch the kind of a drug. So even with respect to diseases using this database. So now I just typed a compound, a drug called aspirin. Okay, immediately I got so many kind of structures. Not only I got the aspirin, but also I got derivatives of aspirin. Okay, so if I want to download these kind of a derivatives, okay, even I can use by clicking this hyperlink. If you see this kind of database. Okay, so you got nearly 121 results for the aspirin. Okay, now I'm taking you to the number one, which is directly taking you to the aspirin, not a derivative of the compound. Okay, if you see this compound, okay, each and every compounds also have a unique PubChem ID. Like how I mentioned, for a nucleotide, you have a GenBank accession number. Okay, and sometimes you also find GenInfo ID. And for the protein sequence, that is for the GenPep, you have GenPep accession number. And for the PubChem compound, okay, that is the natural or synthetic compounds deposited in this repository, you find a unique ID. Okay, so this is an ID you can able to find. And of course, you find IPAC name and physical chemical properties of the compound. Okay, and what are the different names and generic names? So all these things are available. So these are fully annotated database. If you don't know anything about a compound and a drug, but if you want to do a research up to a PhD level or PhDF level, So if you want to know more about the biological information and a kind of a inhibitor, okay, so all this information can be retrieved here. So basically, we go for PubChem to download the 2D structure or a 3D structure. And rarely we go for downloading the 2D structure, but we can use the 3D structure for molecular docking as well as for the MD simulation of ligand receptor interactions. Okay, so if you want to download the 3D structure, okay, you have to take up to the 3D conformer. Now click the download coordinates. If you click the download coordinates, you find the four different formats to download it. Okay, the one which showed at the top, that is SDF. We call this as structure data format. Okay, this format you generally use it for downloading. Okay, so this is what I'll be taking you to how to download the structure and how to go for talking and other kind of natural other kind of concepts. Okay, so generally, if you want to download this 3D structure, I simply click say STF. Don't try to use this kind of a 2D coordinates. Even in 2D coordinates, you get the similar kind of a data formats. Don't download this because it's very difficult for you to put in 3D environment and to go for visualization as well as for molecular docking purposes. So if you see this kind of a compound aspirin, okay, now I'm going to, if you want to edit any content in the aspirin generally, we won't do that. But if you want to make understand about the coordinates, okay, better use a coordinate editor called WordPad. Okay, so generally we go for WordPad to see the contents. So I told you for editing the FASTA, we better use the Notepad. And for editing, or if you want to see the coordinates of 3D structure of a compounds as well as 3D structure of a protein molecules, better to use this WordPad editor from Microsoft Word. So I'm not going to take you to the Linux operating system. Okay, so whatever I'm hands-on experience and the other information I'm going to provide is only from the Windows. So I think many of the everyone have this kind of an operating system. So and also I'm not going to take even commercial softwares. Okay, so I'm going to take one of the freewares from the first class to the 14th class. So I think it's possible with you to practice anything in your desktop computer or in your laptop. Okay, this is how it coordinates. Okay, so on the 3D structure, the coordinates looks like this. Okay, good. Okay, now I just take you to the next kind of database available in PubChem. Okay, I take you to the next database. So the next database is the PubMed. Okay, if you want to download any articles, if you want to download any articles, okay, related to the journal publications, you can either type the PubMed ID or you can type any keyword. Okay, if you want to download any literatures related to a compound or a protein. Okay, so I simply just type the keyword and searching in the PubMed. So you're getting so many kind of PubMed articles. So PubMed articles are high quality articles. Okay, so like generally, if you want to reference any kind of researchers, it's better to go to the PubMed Central for the biotech or biomedical literatures instead of going to Google Scholar. So there are so many kind of databases and plenty of data. Big data are populated in this kind of a database. So everyone try to choose this kind of a database for downloading, uh, even the cancer data. Okay, can be a nucleotide, protein, or gene expression data. All these data are available, publicly available here, including the drugs as well as the literatures available for the cancer research. So now not only we have this kind of an NCBI from US government, we also have another public domain database called UniProt. Okay, so this is another database, UniProt. So in UniProt channel, we call this as Universal Protein database. And here we have plenty of databases like how you find in NCBA. Here, the first database what we find in UniProt is the UniProt KB. Okay, so that is Universal Protein Knowledge Base. So if you don't know anything about a specific protein, for example, I'm working on a protein called TRAP1. If you want to know more about the TRAP1, what is TRAP1, what is the expansion, what is it for? Okay, so the TRAP1 is nothing but a heat shock protein 75. Okay, and this is the molecular weight, and so each and every protein, if you observe, you have a 6-letter kind of a uniprotide. If you see this ID, okay, so for all the proteins, okay, you find the 6 letters, okay, and the first is the alphabet, the remaining five is the numericals, okay. And if you want to get a detailed annotation, you can find the protein information from this. Not only this database is used to get the protein information, but you can also link with the genomic coordinates. So you can also get the information on the genes, chromosomes, and how it is, even the locus of the specific gene in the genome, that is with respect to chromosome, and even the publications related to it. And so all these things also we can link with it. Okay, so what I mean is this is not just a protein, but also linked with the gene, genome, and protein-protein interactions. Plenty of databases are interlinked in this kind of a database. Okay, so we have plenty of information here in this all database called Uniprot. This is a database hosted by London, but also two more countries are also associated with it to develop this kind of a database. And there is one database called UniPark, that is Uniprotein Archive. If you want to download a FASTA format of a protein, better we use this kind of a database. Similarly, if you want to download a cluster of proteins related to a protein family, okay, better we can uh, type the protein family, or you can, if you type a UniRFID, obviously get a list of proteins, okay, downloaded in a single file. And not only that, we also have a database repository for proteomes, taxonomy, and human diseases. So these are few databases, okay, present in UniProt. Basically, we go for NCBI for downloading the sequences, and for fully annotations, better we go for UniProt, because we don't find much annotations here in GenBank. Okay, so if you go to the GenBank or GenPep, get the sequence information alone. Okay, you just get the sequence information alone. I think I've already showed you. Okay, but if you want to get more information, better go for this kind of a database. Okay, now I have taken you to the common biological databases. Now I take you to the PowerPoint and take you to the confined databases for the cancer research. The first database what I'm going to take you to is mycancergenome.org. Okay, so you can find almost all the genes for all the types of cancer in this kind of a large database. Okay, so I just typed a keyword called hepatocellular carcinoma and found the significant, not only get the significant genes of a particular cancer, but also get how much expression it is. Like if you're focusing on a particular cancer, and if you want to find which is the major gene responsible for causing the cancer, even the percentage of occurrence, okay, in humans can also, these kind of data also available in this kind of a my cancer genome.org. Of course, this is one of the important database to determine which is the most significant gene. And the second database, what I'm taking you to, is specific hepatocellular carcinoma database is TBMHCC. Okay, so like this, if even if you go for the ovarian cancer, I also worked on ovarian cancer, kidney cancer. So for all this, we have unique kind of a database for the specific organ. Okay, whatever I target. So it's such a kind of a database for hepatocellular carcinoma, that is uniquely to the hepatocellular carcinoma is TBMHCC. Here you have only the genes pertaining to the hepatocellular carcinoma, that is linked with the liver. And if you see this another third database, oncodb.hcc, this is also a specific database for hepatocellular carcinoma. Simply have to type a keyword okay, of a particular gene, and you can find detailed information on a particular gene on that. Okay. So each and every database is developed from a different university, and of course, even the location varies. And this is another database called cancer liver, that is related to cancer gene expression database. This is developed from Indian scientist. Okay, so that is from the Raghava Labs. Okay, so he has posted a kind of a database. Okay, for liver cancer. So you can even not only can find the information, but also can download the potential biomarkers in the form of Excel or in the form of a CSV or PDF formats. These are large universal database for the cancer genome, that C Bioportal, the most important database, like our you find in CBA for the nucleotide and protein sequences. So if you are doing any research on the cancer, especially you have to focus on the cancer genomics, isn't it? So the first database we focus is the C Bioportal. So here, not only you can find a detailed information, and you have plenty of graph analysis for the sample data sets uploaded in this database. Okay, so each and every data set you can find nearly approximately 100 sample patients information is provided, so their mRNA expression and what kind of a genes, or most important, if you see again here, I mentioned you TP53 is the most frequently observed uh, uh, gene, okay, significant change for that. And not only that, you find more mutations on that, near 118 mutations you can observe in the TP53 gene. And also you can find the survival rate, hotspot, everything. Okay, that is 5 year survival information. All this information related, you can able to find in CBio portal. And there is one more database called OMIM. This is online mentoring inheritance in man. Like the hepatocellular carcinoma, a kind of a liver cancer, is not just caused because of the carcinogens, or it can be from the viruses like hepatitis B or C, but also can be heritable, uh, inherited. Okay, so if so, then what are the major genes? Okay, so based on the population data, we can also find some information on that in this website called OMIM. Okay, that is online Mendelian inheritance in man. And not only for the hepatocellular carcinoma, for any type of a cancer, if you're working on genetics, okay, related to heredity, so of course you have to choose this kind of a website for finding the genes in your research. And the next database, what I'm do is a largest public domain database related to genomic data. Okay, so this is not this is related to, you can find more about the cancer metastasis information. That is, this information can be downloaded from portal.gdc.cancer.gov. So nearly this much projects, and these are the primary sites. So you can able to find the primary sites here. Okay, this kind of a data is pertaining to the world. Okay, mostly from US population. Okay, even we can able to find the Indian population, and we can find the gene specific to the Indian population, which is the most favored gene, favored target for the specific type of cancer. Even we have specific proteins, especially I told you IIT from Raghavan Labs. Okay, so this is actually related to the world information, okay, world data information. So if you want to find more mutations and the other data, you can use that cancer genome portal. And this is a database from NCBI. So NCBI is the largest web resource. We also have a database called Gene Expression Omnibus Profiles. If you type even the type of a cancer as a keyword, and if you search, obviously you get the detailed information on the gene expression, okay, from population data sets. Okay, so you can even go to the specific hyperlink, nearly how much data is that? We got nearly 530,000. So this I've taken some 4 months before. Now, if you will be getting more than you will be getting up to 6 lakhs kind of a data, gene expression data originated from micro. And there's a database called TCMID. This is for a traditional Chinese medicine information database. For this, you can use the CRL megabionet.org/TCMID. So, like how I showed you the receptor target databases. Now I'm showing you the compound, okay, the cancer drug databases. So for the first one, what I shown is from the Chinese database. and this is a Chinese medicine system pharmacology database. If you see the databases for the cancer research, whatever we have in the world, if you exclude China, okay, so that is just a 50% of the databases developed from the China. That is, China is having their own kind of a databases for every type of a cancer, and they have plenty of databases for the compounds, drugs, etc. Okay, so this is one such a kind of database for finding the information of the drugs for a specific disease. And this also the largest database on genes information, gene cards. So if you're a layman, if you don't know anything about a specific gene, but if your supervisor is asking you to work on a specific gene, related to cancer or related to any other diseases, okay, if you want to get more information of a particular gene, okay, just type a keyword called hepatocellular carcinoma, type of a cancer here. I got almost 8,100 genes information, not as even 100 or 1,000, but 8,100 genes information I got. Okay, so not only I'll be getting the genes information description, I get detailed annotation of the genes. Okay, if you click this plus and if you get inside, you get much information, not only with respect to one cancer, but other cancers and also other diseases. How this gene is linked to not with cancer and other diseases. All these detailed annotations also available in gene cards. So that is also a largest database for genes. And TCPA, okay, this is a cancer proteome atlas. So initially I showed you the genome kind of information, isn't it? Genome data common portal, the cancer genome atlas. Similarly, we also have the cancer protein atlas, proteome atlas. TCGA for the genome atlas, and this is TCPA for the proteome atlas. Okay, so for the TCPA portal atlas, you can find the fields like you can select a specific organ. Okay, like what disease if a person is infected from a liver disease, not specifically a cancer. Even you can find the liver disease information from the and also get you can also select a type of a networks. Okay, so the one such network I selected is the bind, but we have plenty of networks. How a protein is interconnected in the proteins to have a link. Okay, even if you want to target a cancer, okay, better you have to target not only a specific protein but also a protein linked with that proteins. Okay, so all this information I get from the proteome, that is protein-protein interaction. And now we can see the open targets. This is another database. Okay, in this open targets, we will be getting a specific target information. For example, I'm working on heat shock proteins. I just typed a keyword called heat shock HSP90. So in HSP90, we have plenty of genes. I selected HSP90A1. And if you want to know more information on that, okay, on the particular target, okay, particular protein. If you see the HS90 protein, you have plenty of genes. But if you want to find a particular gene information of that, okay, of a specific protein, you can search in open target. This is a systematic drug identification. So not only you can get the information on the percentage of expressions, but also get the drugs, okay, for the specific gene targets in that database. Now I take you to the pathway database. The first pathway database, what I'm going to explain you is on KEGG, KEGG, Kyoto Encyclopedia of Gene and Genomes. This is the largest pathway database where you can find the for metabolites. We can also find for diseases. Almost we can find all the biological pathways, okay, in this KEGG. Actually, KEGG is developed from Japan. Okay, so we have a URL accordingly, like www.kegg.jp. Okay, so here now initially just showing you the hepatocellular carcinoma pathway database. Okay, so related to the association of the disease. Okay, what are the important target proteins and what kind of symptoms the hepatocellular carcinoma patients face. Okay, so all these symptoms linked with the protein interactions, or all these things are shown in this path. And similarly, one more server called GEPIA. This is again, once again, related to the gene expression. Okay, if you want to find genes present, okay, the differential gene expression present on a particular organ. If you want to find a particular gene that is HSP90A1 in liver, how about, is that a significant or not? If that is a significant, what are the genes linked with it? That we can get from this database. Not only we can get for a single organ, if you want to work on pulmonary disease, if you want to work on the lungs, okay, if I want to check for the lungs whether HSP90A1 is significant or not, even I can locate the specific organ and I also check the interacting proteins. It doesn't mean that whatever proteins linked with HSP90A1 liver is also linked with lung. For the lung organ, uh, protein interactions fail. Okay, so all this typical kind of a protein-protein interactions related to a specific organ can be found from here. Not only that, we also find the tumor versus normal. Okay, that is, you can find these two types of data in multiple samples in a data set. Okay, so you have also plenty of data sets. So you can take a data set and you can find how much tumor and how much normal samples, and you can work on the analysis and you can find the significant change. Okay, so all these things are represented in that database. And now we can go to the next server called Enrich-R. So this is meant for gene enrichment database. So in my publications, you can see latest publishing scientific reports, informatics in medicine unlocked. So in these publications, I just using this kind of gene enrichment. That is, if I want to find a most significant gene for a particular cancer, better I go for initially a top. I identify the top 100 genes, and then I limit up to the top 10. Initially, I go for the top 1,000, that is top 500 upregulated and top 500 downregulated genes. And then I go to the top 100 genes. That is, I'm going to screen it, okay, to the top 100, which is more significant based on the scores. And then based on the p-value, then I bring it down to the 10 important genes. And then I just use this 10 genes as a therapeutic targets, which interacts with the hub gene called HSP90A1 in my case. So likewise, okay, so we can able to use the gene enrichment using Enrich-R. And there is one database called String. Okay, String database is for a protein-protein interaction database. This is the largest database where you can find protein-protein interactions. That is almost 67.6 million interactions you can able to find. The interactions is around 20 billion, but this much number of proteins are there. Okay, 67.6 proteins are there, fetched from 14,094 organisms. And in the String database, okay, how these data are deposited. So only through the wet lab techniques, isn't it? So what are the type of wet lab techniques and how these data are derived? All these informations are present in this kind of a database. Okay, so the annotations and the conclusions derived from the protein-protein interactions can be studied from the String database. So one such uh, output shown from the string, simply I'm typing a specific protein, whatever protein I'm going to type, okay, you can find the interacting proteins in there. You better try with the TP53, which is the most significant gene, not only for the liver cancer, but also for the other cancers. So when you search in any databases, okay, better search using the TP53, you can find more significant information when compared to the any other genes. And functional enrichment of the string database. So even it can enrich, like here, whatever you are getting, okay, is a top 10 interacting proteins of HSP90A1. Okay, but if you want to uh, check the top 50 interactions or top 100 interactions, everything is possible. And even if you want to filter the data based on the enrichment, that's what we call as a functional enrichment. This can be done for the biological process, molecular function, cellular component, check pathways, reactome pathways. Okay, so if you're focusing on a particular molecular pathways of a particular protein, so you have to select the pathways. Then you can also select the tissue which you want to target. Then it can able to get the more appropriate kind of networks, protein-protein interaction networks connected to the hub protein. So such a thing, I made it for a kind of liver cancer. Okay, so nearly, I just checked the liver, the string database. Okay, so this is the initially whatever I shown is the HSP90A1 connected proteins. But now when I selected the liver, you can see only the liver organ associated protein-protein interactions alone are shown in red color. The remaining cyan color is not considered, which is not important for the liver disease. If you want to target this as an important target, that is HSP90 as an important target for a liver, then obviously you have to target only 1, 2, 3, 4, 5. Only these 5 proteins along with the HSP90. Similarly, if you want to go for a functional enrichment with other kind of filters, okay, even that can be made possible. Initially, I tried with the liver, which I found it selected in the red color, but also when I want to try with the hepatocellular carcinoma cancer, as the second functional enrichment, I selected the hepatocellular carcinoma, which I think hepatocellular carcinoma is shown in red color, and the liver, what I selected is shown in the blue color. So obviously, the blue and red are the more significant interacting proteins, and the cyan color proteins, even though it is connected with the HSP90A1, but these are neglected. Okay, these are not considered as important targets. When I checked even the expression patterns, okay, so the interacting score, I can able to get these are the 5 important protein targets for HSP90A1. And similarly, there is one more database called Stitch. Okay, in Stitch, not you know not only going to find the protein-protein interactions, but you also can find the drug-protein interaction. So there's an additional feature available in this database called Stitch. Earlier, whatever shown is the string. This is a string database, and this is the Stitch database. You can able to find plant compound with RNA, which is interacting with the different proteins in the Stitch database. And even here, you can do the functional enrichments, and you can select the important protein targets. Okay, so even you can download these kind of networks to the cytoscape, and you can visualize all these things, and you can do the functional enrichments, and because as it's the data, whatever download from the database can't be taken for the publications. If you want to publish these kind of data, better take it to the site, take to the cytoscape, and you can go for any kind of enrichments, and then you take this for the publication. So this is something about oncology database. So I think almost I have taken additional 10 minutes because we are the first class, we had an introduction. Okay, I have taken 10 more minutes extra time to complete about the oncology databases. I think the MATLAB is uncovered on today, but it will be accumulated in the subsequent classes. I will be trying to complete the MATLAB in wherever we find gaps in this next subsequent classes. And participants, my suggestion is to just join the session on time because it disturbs others if you're not accepting the invitation. Okay. And if you have any doubts, you can ask me. So I've completed my session, the first day session. If you have any doubts, you can ask. I can clear 3 doubts on each and every day. Anyone have doubts? Yeah, can you show us a demo on how to use the string data? Yeah, okay, I can show you. So I just going to the string, simply have to type string db, okay, in the Google. It takes you to the string -db.org. So this is the functional protein association networks. So here, this is the updated information. Nearly 20 billion interactions, 59.3 proteins are there, and totally this much number of species. Simply click search. Again, I'm showing you once again. Coming back and showing you once again. So initially, you have one kind of string-db.org as the URL. Go to the string, then you get this kind of a window. So either you type the protein name, okay, or if you want to search with respect to the organism, choose the organism. It is better to type through the protein name and finding the interaction of the specific protein. So if I want to find for the TP53, I'm simply typing TP53. So if you don't find any chain, okay, you check for some other related genes. Okay, I can't guarantee that all the genes are shown here, but whatever is posted Okay, so that information, that interactions information alone is shown here. So when I search for a TP53, okay, default it takes you to the multiple species, nearly 1,794 species information is available, but no, not just the 1,794, but also related proteins. Okay, but I want to take only the TP53 because the human. So default it is selected. So simply click continue, get inside. So defaultly Homo sapiens TP53 is selected. And not only the first session, in all the remaining 13 sessions, I'm going to take human as the subject. Okay, so that is, I'll be taking only human species, and I'm going to take mostly on the cancer research studies because our theme is pertaining to the cancer, isn't it? But if the cancer, this kind of a cancer, you can also find hepatitis B and C or the causative agents. Under that conditions, even I'll be taking a viral etiology. Okay, so I generally focus on viral and cancer diseases in my research, especially target only the humans in bioinformatics approach. Okay, so simply what I did, I just selected the default one. Simply I have to click continue. After such sometime you'll be getting a kind of a protein interaction networks. Just because of network, okay, traffic, the network traffic, I think we're getting results delayed. Yeah, now almost I think it's getting loaded. So there's a protein interaction network for the TP53. We can do much with this protein interaction networks, all this detailed information. Okay, so we'll be discussing on ninth session, network pharmacology and pathway analysis. Okay, on ninth session, I'll be taking you more on that. Any other doubts you have? Sir, which one is the best to check the interaction between protein and some other compounds? Stitch, stitch TB. Like string TB, you can better go for the stitch TB. Stitch gives information on reported protein, dark protein. Yes, you are right. You're right. Good. I think I have cleared your doubts on the first day session. Okay, participants join on time. And see here, don't uh, come like this. Okay, for each and every time if I try to admit, it disturb you and I reduce teaching. Okay, so to join on time and accept the invitation here. Thank you. Have a great day to you.
Thank,
Thank you,
thank
sir.
you, sir.
Thank you,
Thank
sir.
you,
So
sir.
I thank you all the participants who
Thank you.
interacted and